All right, let's dive right in. In this explainer, we're going to crack open the genetic code of cancer itself, and we're going to focus on one of the absolute central players in this whole story, the oncogene.
So, let me ask you something. Just imagine for a second that every cell in your body has a gas pedal and a brake, to control when it grows, when it divides. It's this perfectly tuned system. But what if that gas pedal got stuck? I mean, really stuck, jammed right to the floor. That's the question we're tackling, because that's pretty much exactly what an oncogene does to a cell.
Okay, but before we get to the villain of the story, and oh, there's a villain, we have to meet the hero. We've got to talk about the good guy first. See, these are the genes that, when they're working normally, are absolutely essential for us to even be alive. These are the proto-oncogenes.
Yep, proto-oncogenes. These are the good guys. They're the ones that give the green light for a cell to grow and divide. But, and this is the key part, only when it's supposed to. Think of them like super responsible managers, making sure new cells are made when you need them, for healing, for normal growth. They're the carefully controlled go signal.
And this really breaks it down, doesn't it? The proto-oncogene, that's our hero. It's the gas pedal you use carefully, capping it just right to get where you're going. But when that gene mutates, it becomes an oncogene. That's our villain. Now the gas pedal is stuck to the floor, and you've got this relentless, uncontrolled, cancerous growth.
So this is the big turning point in our story. How in the world does this essential, heroic gene go so wrong? How does it get corrupted? Well, the answer is pretty simple and pretty scary. It's all about genetic damage, a mutation that transforms our hero into a total villain.
So yeah, here's the formal definition. But what's really important to get is that an oncogene isn't some virus or bacteria invading from the outside. No. It's a corrupted version of one of our own genes. And that corruption, whether it's from a mutation or just having way too many copies of it, that's what gives a cell that non-stop command to grow. And that, right there, is a hallmark of cancer.
Okay, so what are these corrupted genes actually doing inside the cell? Well, they basically hijack the entire control panel. They can act as something called transcription factors, which pretty much force the cell to keep reading the grow and divide chapter of its instruction manual over and over and over again. They also crank up all the pro-growth signals, basically shouting, "Divide!" nonstop. And maybe most importantly, they shut down the cell's own self-destruct program, a process called apoptosis. This lets these totally abnormal cells survive when they should have been taken out of the system.
Right, so this transformation from hero to villain, it doesn't just happen randomly. It happens through very specific kinds of genetic mistakes. So let's break down the three main ways a proto-oncogene gets corrupted. All right, here are the three big ones. You've got simple typos in the DNA, you've got these major cut and paste errors, and you have a genetic photocopier that's just gone completely haywire.
First up, let's talk about the point mutation. And this is wild. It's like a single-letter typo in the billions, literally billions of letters that make up your DNA. A tiny mistake. But, and here's the kicker, if that typo happens in just the right spot in a proto-oncogene, it can permanently jam the protein that gene makes into the on position. A tiny change with absolutely catastrophic results.
And just how big of a deal can one little typo be? Well, look at this number.
Yeah, that's right. A staggering 90% of pancreatic cancers are driven by a point mutation in just one proto-oncogene, a gene called KRAS. Think about that. One tiny error is the main engine behind the vast majority of cases of one of the deadliest cancers we know.
Okay, next mechanism, gene rearrangement. The best way to think about this is like a really bad corporate merger, but happening at the genetic level. A piece of one chromosome literally breaks off and gets stuck onto another one. And this doesn't just move the gene, it can create a totally new hybrid supergene with dangerous new powers. The classic textbook example of this is something called the Philadelphia chromosome. In a type of cancer called chronic myeloid leukemia, or CML, a piece of chromosome 9 breaks off and fuses with chromosome 22. This mashup creates a brand new fusion gene called BCR-ABL, and this new gene produces this hyperactive protein that just constantly screams at cells to divide. This one event is literally the defining cause of CML.
All right, our third and final mechanism is called gene amplification. Now, this one is less about changing the message and more about just screaming it at the top of its lungs. The cell's machinery basically messes up and starts acting like a photocopier gone wild, making dozens, sometimes hundreds of extra copies of a single proto-oncogene.
Just look at the proto-oncogene HER2. A normal, healthy breast cell has exactly 2 copies. But in a huge chunk of breast cancers, up to 30%, that gene gets amplified like crazy. The cancer cell can have over 50 copies. Can you imagine? All those extra genes just create this flood of growth signals, which is what makes the cancer so aggressive.
So now that we know how these genes go bad, let's meet a few of the most notorious villains in the cancer story. These are the specific oncogenes that scientists and doctors are hunting for in patients every single day.
And here's our rogues gallery, connecting the specific gene to the specific crime. We've already talked about HR2 in breast cancer and BCR-ABL in CML. But the list goes on. You've got EGFR, which is a major driver in a lot of lung cancers. You have MYOC, which is famously switched on in Burkitt's lymphoma. Each one of these is a specific target, a specific way that the normal process of cell growth gets totally hijacked.
Okay, I know all of this sounds pretty grim, but this is where the story takes a really hopeful turn. Because understanding exactly how these genes go bad, well, that's turned out to be the single most powerful tool we have in the fight against cancer.
It all comes down to this one simple idea. By identifying the villain, we can design the weapon. And let me tell you, this knowledge has sparked a revolution in how we treat cancer. We now have what are called targeted therapies. These are drugs designed specifically to find and shut down the rogue proteins that these oncogenes are making. So we're not just carpet bombing all fast-growing cells anymore. We're taking aim directly at the genetic heart of the cancer.
And that leaves us with this one final thought to chew on. We've found so many of cancer's critical on switches. We've even figured out how to flip some of them back off. Now, the challenge ahead is still huge, of course, but the path forward is clearer than it has ever been. So the real question is, what's it going to take? What are the breakthroughs that will finally let us silence them all for good?